Okay. I'm Alexei Sharov, and today I will present my short slides about semiotic agents, which are evolving, learning, autonomous, and goal-directed entities. I am a biologist. Originally, I was trained as an ecologist and entomologist, but then I switched to molecular biology. So I covered kind of various areas of biology, but all my life I was interested in theoretical biology from my student years. And initially we had a working group on theoretical biology, but then it was closed by administration, and later in the late 80s, I decided to revive this tradition and organize my own seminar on theoretical biology. And I was looking for a good title for this seminar because I wanted to have a more specific title, not just general theoretical biology. And I came to the term biosemiotics, which is a combination of biology with semiotics, which is a study of signs and meanings. So in 1988, I organized a regular seminar every week, and then I also organized two winter schools on biosemiotics, attended by about 100 people in Russia. And that was the beginning of my studying of biosemiotics. Later, I realized that the term biosemiotics has been coined already before me. So it was first proposed by Friedrich Rothschild, and then also used by Stepanov, Thomas Sibiok, Kalevi-Kuhl. So several people were using this term, so I was kind of joining the same tradition. So everybody actually was inventing this term independently. So it was basically flying in the air, and people came to this idea. So the idea was to go a little bit away from cybernetics into semiotics. So why this tradition was important? Because life is meaning making, and so it's not just information, because information is just a carriers of life. So it's a tool for processing meanings, and we need to go to the meanings themselves. And so I consider biosemiotics as a kind of equivalent of theoretical biology. And at some point, I realized that the central notion of biosemiotics should be not a sign as in semiotics. So semiotics was mostly a humanities discipline, so it was looking for signs, meanings, different kinds of signs. But what is important in its application to biology is agency. So agency as the central unit that processes signs and involves in activity related to signification and extraction of meanings, and using these meanings for living functions. So it was more pragmatic understanding of semiotics. So this pragmatic understanding of semiotics goes back to Charles Morris. So Charles Morris was the follower of Charles Peirce, who is considered the classics in semiotics. But Charles Morris was focusing mostly on pragmatics. So he thought that pragmatics, and especially the use of science by humans and animals, is an important part of semiotics. And so biosemiotics came out of this tradition. So it was started from basically Jacob von Uexküll in the 20s and 30s of the 20th century. And so he wrote a book on the meaning of science in nature, Bedottens Lehre. So he didn't use the term biosemiotics, but basically he was talking about biosemiotics. And then this tradition was taken by Thomas Sebeok in the United States, who first developed zoosemiotics, but then he realized that he needs a broader scope, so he also came to the term biosemiotics. So, of course, the big support for biosemiotics was the understanding of the genetic code, so the realization that some kind of language is behind life at the very molecular level of life. So even there we see something like a language with its syntax, with its partitioning into meaningful segments like genes. So that was a great stimulus. And it was important to understand what is the relation between this molecular level of sign processes and animal behavior and communication. Okay, so this is the title, Semiotic Agents, Evolving, Learning, Autonomous, and Goal-Directed Entities. And what I would like to start with is what I call the digital paradigm, which is a prevalent way of thinking in biology and related fields. What it does is that the initial disturbance or signal that moves a system away from the equilibrium is declared a cause of change, whereas the network of interacting factors that control the subsequent dynamics of the system, which basically is interpretation of these signals, of these perceptions, and controls the subsequent dynamics of the system, is ignored. So there is direct link between the signal and the response without any intermediate processing. So to understand the living system, it is important to take into account this intermediate processing, not just simple physical cause-effect relationship, but also complex semiotic causation. So example of semiotic causation is in human life. When the traffic light turns red, the driver stops the car. So this is a very complex event that is multi-stage, and it's very consistent. Nevertheless, the freedom is retained in this relation. So if something else happens, so for example, if you are chased by a dangerous person who wants to kill you, you probably ignore the red sign and go forward no matter what. Or if you are trying to escape a tsunami, you will also drive through the red light no matter what happens. So this is a more complex causation that is needed to be considered in biology. So here is the pairwise comparison of the mechanistic biology and the analysis of semiotic agency, which is a core notion in biosemiotics. So the mechanistic biology is focused on efficient causation, on the mechanism, whereas biosemiotics and the analysis of agency is focused on a final causation, teleology, and formal causation, which is signification. In mechanistic biology, biological function is just a process, and goals are externally defined. So this is a very popular now notion of process ontology. So people think that they discovered a new area because they moved from individual objects to processes and think that this is the main achievement. However, you need to move further to semiotic agency and see how biological functions work as means to solve problems by the agency. And then goals are not externally defined anymore. They are emerging within the agent. And so that is the way of understanding life and cognition. Then the next point is a mechanistic view considers each process at each time and spatial scale as a separate entity and try to model it as a separate unit. Whereas for agential approach, agency is analyzed at multiple scales in time and space and with all its functions and all its subagents. So, for example, in a multicellular organism functions not as a just undividable unit, but also as a collection of cells, collection of organs, and how they function. So some functions are delegated to subagents. Then if we look at the environment, for mechanistic view, it's just a boundary condition and disturbance. For the agential view, a local environment and tools appear to be a part of agency. And that's why niche construction appears to be so important for evolution and for ecology of organisms. The next point, in mechanistic biology, try to emphasize similarity of processes at various scales, and quantitative differences are downplayed. So that concerns, for example, the notion of life. So mechanistic biology try to see that there is no boundary between physical processes and living processes. That's why the uniqueness of the phenomenon of life becomes lost. The same thing happens with cognition, with learning. So these terms are expanded to the whole tree of life. So people say that even bacteria are capable of learning, they are capable of cognition. And so that diminishes the importance of these terms at the higher levels. And this terminology problem kind of devaluates the uniqueness of living phenomena at different scales. Whereas then a study of semiotic agency emphasizes qualitative changes in evolution, development, and behavior. And this is important to show the creativity of life, that really novel entities are emerging at each step of the evolution or learning. So this creative view of life comes back to the famous book, Creative Evolution, by Henri Bergson. So that's the following of this tradition of creative evolution. Then mechanistic biology use wide extension of meanings of well-established biological terms. So this is basically almost the same that I said before, whereas semiotic agency introduces semiotic terms into biology with a wider meaning. So the terms of agency, sign, goal, and semiosis. So instead of saying that bacteria are learning, we say that bacteria have sign processes or semiosis. And so they are capable of interpreting signs and capable of creating new signs in the lineages of bacteria. Okay. So the definition of semiotic agency that is used in our book, written by me and Morten Tonneson in 21, is that agency is the capacity of acting purposefully and using science to make informed choices. So purposefulness or teleology is a key feature of agency. And this is what is not covered by physical approaches or cybernetic approaches. So teleology is an important thing here. Signs are carriers of meaning for certain agents. So signs are not a kind of isolated entities that have their own properties. They have sign properties in relation to certain agents. So this is kind of relational ontology, not essential ontology like in physics. So in physics, we look at some object and say the essence of this object is such and such. With science, it's not like that. So any physical object can be a sign for certain agents. So it is a relational ontology. And signs are mostly passive in terms that they are not actively changing agents on themselves. Instead, agents are actively interpreting signs and act accordingly. So agents is the active component, and agents are more important than signs because it is the agents that establish sign relations and modify them as needed. So this is a kind of switch in emphasis in comparison to traditional semiotics, because traditional semiotics is focused on the signs themselves, and semioticians, they try to think of signs as special entities on their own. However, this agential approach puts the emphasis on the agency, which is different. So agents can act adaptively even with incomplete information without knowing mechanisms and physical causes. So that's different from the physical explanation of living phenomena. So in such cases, agents rely on semiotic causation or phenomenology. So they are using their previous experiences, their ability to connect causes with consequences, to plan their activity, even if they don't know the mechanisms. So the notion of agency leads us to reconsidering how the life process proceeds. So the traditional gene-centric idea of life and evolution is emphasizing the informational channel for transfer of hereditary information from one generation to another. So here, agential approach adds the second channel, which is agential channel, which is active. So the information is mostly passive. And it requires interpreters, it requires agents also for copying information, for repairing it, and for transferring it to the next generation. So the agential channel is the physical agency. So it's not just encoded agency. So some people say, oh, all the proteins are encoded in the genome. But for copying the DNA, you need actual physical agents that are already converted into the agential form. And actually, agents are carrying a lot of their own specifics that is transferred to the next generation. So it's not written in the genome. It's just an agency. It's a part of the agency. It's simply transferred, duplicated, augmented, and that's how the life process progresses. So that's why it's this agential part is the most important part of evolution, whereas informational part is simply a tool, an informational semiotic tool for keeping records of the memory, hereditary information, both genomic and epigenomic, So it's a tool for agents, whereas agents is the central thing that is changing. So this is different from the neo-Darwinistic view of evolution as a selfish gene proposed by Richard Dawkins. So although genes are also kind of agents or parts of agents, but the real agents are organisms, and these agents are hierarchically organized. The organisms are composed of cells, or cells are composed of subcellular agents. So this is a much more complex process. So what is actually semiotic agency, and why this term is important? It is important because the notion of organism doesn't cover everything. So it covers only the whole organism, and the selection of this level of organism level as the highest level of organization is sometimes arbitrary. So sometimes you don't know which level of agency is the most important, especially in the transitional cases. So, for example, when eukaryotic cells emerged by semiogenesis of several kinds of bacterial-like organisms, it was not clear which of them will take over eventually and which of them will play minor roles. So it's important to have a term that covers all levels of semiotic agency. So the organisms represent the core example of semiotic agency, but also we need to add organism parts, autonomous organism parts, such as organs, cells, and also superorganisms, such as colonies, ecological consortia, especially those that are closely integrated, like lichen is closely integrated ecological consortia. So there are a lot of examples in ecology where organisms cannot live without each other. So basically they form a united whole, or take what is called a holobiont. So a holobiont is a multicellular organism together with all its microbiota. And it appears that microbiota plays an important role in the physiology and even evolution of these multicellular organisms. So this is important. Interestingly, so I already mentioned that agency needs teleology, but teleology is not always generated within organism lifespan. So teleology can be borrowed from parental organisms, and also parental organisms can kind of program their progeny or their tools in a certain way so that they will act as agents. So agency can have kind of external teleology. And considering this, we can add autonomous tools to the category of semiotic agency. Examples are ribosomes. So ribosome is not a self-sufficient agent, so it needs to be constructed by the cell. The same thing with proteins, protein complexes. So they act as agencies, but they have external teleology. And as for human life, we construct computers, robots, cars, and it's also autonomous tools that are augmenting our agency, but they have their own autonomy as well. And they have external teleology on their own. And also we can add temporary organizations which are typical for humans, such as businesses, governments, nations, and so on. So that's the whole scope of semiotic agency. And of course, you can see that it's a very large and set that includes very different systems by their complexity. So that's why it's important to kind of differentiate what kind of agency is there. And that's why it's important to differentiate different levels of semiotic competence in these agents. And here I, at the top level, differentiate between what is called protosemiosis and eusemiosis. So protosemiosis is a molecular level semiosis, which is based on signal sensing and signal interpretation, like protein receptors are binding the ligands like glucose molecules and responding. So this is protosemiosis, which is signal-based interpretation. So there is no much of integration of signals. So mostly signals are summed up. So similar signals, cells can say that the signal is stronger or weaker. This is possible, but this is still within protosemiosis. When we go to eusemiosis, so eusemiosis is typical for eukaryotic cells, whereas protosemiosis is in bacteria and archaea. In eusemiosis, sensorial patterns are linked with individual objects. So that's how already protists can differentiate between edible and non-edible particles. And later, this capacity is increased in higher eukaryotes, and especially in multicellular eukaryotes that have special sense organs. So detecting objects and recognizing objects of different types, which is called categorization of objects. Then the next step is differentiation within eusemiosis. So here I proposed three major steps. The first is innate eusemiosis, which is instinct-based cognition, which is adjusted by content-dependent plasticity, sensitization, habituation. So lots of animals have this kind of innate eusemiosis. Also plants, fungi, all these organisms have innate eusemiosis. Then consciousness arises only within certain animal species. And it is characterized by true representation-based learning, not just plasticity, and it requires representational memory. So it's especially visual memory is used here as one of the major part of representational memory, but acoustic memory and other also can work as representations. And the final is abstract thinking, which is observed only in higher animals, mammals, birds, and most developed it is in humans. Of course, we can add some additional steps in this process. I kind of just pointing the major transitions. Then an important term in biosemiotics is semiogenesis, which is emergence of new kind of sign relations. So semiogenesis is distinguished from semiosis, which is mostly using of already existing sign relations. Of course, there is a question of how do we determine novelty. So novelty can range from minor novelty to major novelty. And the main feature of novel sign relations is that it is not automatically produced. So if you have just some syntactic rules to how to interpret new signs, it's not a real novelty. So it's basically application of existing algorithms already. So the semiogenesis should involve emergence of some new algorithms of processing. And in cognitive animals, the major source of semiogenesis is learning of new behaviors, concepts, so it's mental models of reality. A few words about semiogenesis in evolution, because now the dominating concept, evolutionary concept, is neo-Darwinism. And it lacks some important components of evolution. According to neo-Darwinism, there are new random beneficial mutations that then picked up by natural selection, and it becomes the part of the new form or new behavior and so on. However, it appears that there are no new random beneficial mutations in nature. Instead, some mutations, so even all mutations are disturbances, but some of them can be interpreted creatively by cells and or their subagents based on their innate adaptive competence, and that may have beneficial effects. So the idea is that it is not mutations itself that is beneficial, but its interpretation by agency. And agency become trained in interpreting errors, because only that will ensure reliability and robustness. So instead of new random beneficial mutations, biosemiotics accepts the process that is called genetic accommodation. So that means that adaptations, they emerge first by the activity of agency. Of course, they are not always well tuned, but they will need some additional tuning, and they will need automation, but this comes later during genetic accommodation. So that's where what is called usually called natural selection takes place. So one of the evidences of that is that all known beneficial mutations in nature are not new. These evolutionary changes have been already well rehearsed in the history of lineages. And I will give you two examples. First is useful mutations. One of the useful mutations is antibiotic resistance of bacteria. So how does it happen that some bacteria are resistant to antibiotics? It appears that resistance of bacteria has been existing long before the invention of antibiotics, but because all bacteria and other single-cell organisms are competing with each other, and they're using chemical welfare against each other. So they need to be adapted to toxic substances. So they already have this habit of overcoming toxicity of external molecules. This habit just needs some minor adjustments through physiological plasticity. Second example is the peppered moth. So it's a famous example in natural selection. So the peppered moth is hiding on the trunk of birch trees, and that's why it has originally white color. So birds cannot see it and do not eat it. However, during Industrial Revolution, there was too much soot in the forest, and the birch trees were covered with soot. They became black, and that's when the black form became abundant in the industrially polluted areas in England. So how did pepper moths manage to produce new coloration so fast? It appears that dark color is not new for ancestors of peppered moths. Most Lepidoptera have dark coloration as a common natural variation. And why do they need it? They first of them, they need to survive on different backgrounds. They need to capture more sunlight in spring, for example. That's why spring forms are usually more colored. So when the British scientists tried to find what genes caused this carbonaria form to emerge, they couldn't find such a gene for a long time. So this lab worked for decades, led by Van Hoff, and they found no association between carbonaria phenotype and any melanin-related genes. Melanin is the pigment that cause dark coloration. But after a while, they found something. So they found that a dark phenotype is associated with a transposon. So that was a big surprise because transposon doesn't code anything. So it's basically a virus. It's a disturbance. So that's, and the authors tried to find some functional roles, but their evidence was extremely weak. So there was a little bit of increase of expression of one gene where this transposon is located, but the effect was very weak and there was no functional validation of this study. So what we can say about this? So here, a transposon was used simply as a disturbance that was interpreted by, so probably, how I interpret it is that probably darker coloration was initial coloration of the ancestors of the pepper moth. And then they adapted to birches, which is white, and they lost melanin in the wings. And probably what this transposon did is simply it restored the original phenotype of their ancestors, which were dark. So that's probably what happened. Of course, this needs to be further research, but this is an interesting example where the search for a useful gene ended nowhere, basically. So why genetic accommodation is much more realistic than the hypothesis of a new random beneficial mutation? Because new beneficial mutation needs to be fast, it needs to be highly specific. Probably it should involve not one nucleotide substitution, but several substitutions and no substitutions in other places of the gene. So it is a very highly unrealistic event, which if it can happen, it would happen in millions of generations. So the organisms should wait millions of generations before such a mutation would appear. Whereas genetic accommodation is not urgent, because organism can already adapt roughly to changed conditions. So the organism can wait a little bit. That's the first factor. And the second factor is that there is no need for specific mutations that changes the protein exactly to a new beneficial form. Most subagents in cells have multiple functions. And besides their main functions, they have additional functions. And usually these additional functions is the playground for developing new adaptations. So there may be additional ways of initiating these additional functions by, for example, combining with different other components in the cell, with different proteins, forming new combinations of proteins or riboproteins. Involvement of these combinations can be initiated by a variety of mutations anywhere in the genome, which are non-specific, and they may concern not only the protein structure, but also protein expression, repressions, lots of effects, degradation, transfer, and anything can affect these additional functions. And these additional functions, that's why they have much higher probability to emerge. And then they can compensate for, they can adjust other functions, and they compensate for the insufficient beneficial effects, which is basically the mechanism of genetic accommodation. Finally, I would like to just mention a few other projects that I was doing in my life. So I was studying the origin of life from agential point of view. So the current vision of the origin of life is exclusively chemical. So they people think that if they have a good combination of molecules, then they have the life. I try to come to this problem from a different point of view. What those primordial systems that were emerging at the origin of life were doing, what kind of problems they were facing to overcome in this hostile world to become alive. What I realized is that the most likely place for life to emerge is the surface of oil droplets in water. Why? Because first is the division between physical phases. So it's beneficial for catalysis because it's two-dimensional. Second, it's liquid because it gives much more flexibility for molecules to move around and interact with each other. Third is that hydrophobic oil droplet can accumulate additional chemicals. In particular, they can accumulate polyaromatic hydrocarbons, which appear to be very highly abundant in some star systems. And they're produced by stars that are rich in carbon atoms. And these polyaromatic hydrocarbons, or PAHs as they are called, can be absorbed by oil droplets, and they can help in catalyzing reactions that involves capturing photons, ultraviolet photons, and for example, oxidizing the surface of the oil droplet. So if the surface is oxidized, carbohydrates are turned into real oils, into try to remember the term. When I'm presenting, it's hard. And this changes the surface tension of the oil droplet, so it becomes more hydrophilic. So it's easy for these droplets to divide. So then some coenzyme-like molecules can appear on these oil droplets and help. They can cooperate with aromatic compounds within the oil droplet, and then it can start oxidizing the surface and form fatty acids. Fatty acids. So this is how the initial catalytic activities can happen on these oil droplets. And then oil droplets will evolve by adding more catalysis, by polymerizing of these catalysts. So from here, from these polymeric PAHs, you can go to something like nucleic acids that will start duplicating with the help of these catalysts. And eventually, some water will be engulfed inside the oil droplet, and this will the surface will become stabilized and form a cell with a membrane. So that's how I envision the emergence of primitive cells. So that's one project that I was involved in. And the second one is the change of genome complexity in macroevolution. So what I did is I was trying to quantify the length of the functional non-redundant genome in various groups of organisms, from prokaryotes to mammals. And of course, in mammals, I had to consider that not the whole genome is functional, but there are lots of non-functional regions there. So I just quantified the functional portion of those genomes. And it appeared that the log length of functional genome is approximately linearly related to the time of origin of these taxonomic groups of organisms. And then this regression goes back in time, and this projection gives me an estimated age of life, which is about 9.7 billion years ago. So that's certainly much younger, much earlier than the appearance of the Earth, which indicates that probably Earth was contaminated with bacteria from space, a phenomenon which is usually called panspermia. Okay, so that's the end of my talk. So semiotic agents are systems capable of acting purposefully and making informed choices. So they belong to a special category of being because their properties correspond to, are self-determined. So they have self-determined identity. And signs are meaning carriers for agents. Semiotic agents vary in the levels of complexity, autonomy, and so on. They have semiogenesis, they can make new signs, and most agents have a composite nature and support reliable self-construction, robustness, and adaptability. So that's basically the end of my presentation. So I will stop sharing the screen.
Super. Yeah, thank you so much. Very interesting. So I have a couple of things to talk about. So I love the emphasis on creative interpretation of existing mechanisms as a road to adaptation to new challenges and so on. I think that's very important. I think that's right on the money. And in general, I'm very sympathetic to the whole kind of semiotic perspective. I have one question in your sort of point, but the spectrum of qualitatively different stages that you had. Why do you think? Can you talk a little bit about why consciousness comes late there, in the sense that you said it requires memory, especially representational memory? I'm curious about your thoughts. Why do you think consciousness requires representational memory?
Mm-hmm. So here, my conclusion is based on the books of Yablonka and Ginsburg. Ginsburg or Yablonka, The Sensitive Soul. It's a very fundamental manuscript, and they showed that unlimited associated learning, what they called unlimited associated learning, comes relatively late. And it certainly requires a well-organized nervous system. So this should be either brain or a very highly integrated ganglia, like in some mollusks, in insects. So it's kind of brain-like structures. And they can support this unlimited associated learning. What they call limited associated learning, it's basically it's not learning at all. So I think it's better characterized as plasticity, because it's basically plasticity of neurons. So the synapse becomes plastic, and it can be either sensitized or repressed. So it's very non-specific. Whereas unlimited associative learning is highly specific, because it's based not on one neural pathway, but it depends on a large number of pathways which are integrated together. So they are reinforcing each other. And that's why it becomes very specific.
Yeah, I understand. It's an amazing book. I agree with that. I'm sort of thinking even more broadly, so beyond neurons at all, at the wide range of potential mechanisms that could underlie the algorithm that you're talking about, right? So associative learning, unlimited associative learning could in principle be instantiated in all sorts of things that are not neurons, not biologicals even.
Yeah.
And so I'm just interested which of those kinds of functional components are thought to underlie consciousness. And I say, I can imagine a kind of consciousness which is very with zero memory, right? That is just a perception of an inner perspective that is at every moment has some sort of state that, it could be stress, it could be, whatever it could be, but there's sort of like, it's a very primitive kind of consciousness because it has little continuity, no, but you can sort of imagine that, right?
Yeah, so you're using again this what I called a mechanistic approach, where you emphasize similarity more than differences. So I'm trying to emphasize differences or transitions, so kind of fundamental transitions in evolution and functionality.
Yeah.
And if we apply all the terms too early, then we run out of good terms, because this is our language that we're speaking about, and we should be careful to keep enough terms for the upper levels. If we apply them all at the early stages, then we need to invent something extraordinary at the top.
Yeah. Yeah, no, I agree with that, and I think the terminology needs to keep up with the science, and my suspicion is we may well need new terminology. Language is an important one, because while there's a lot of work, I think, on in the diverse intelligence field on many of these phenomena outside of the typical kinds of animals that we're talking about, language seems to be like proper language. I have not seen any evidence for proper language in any of those things. So that seems like a good, high level term, and we may need other high level terms. One other thing that I wanted to mention. So at the end, you mentioned this panspermia idea. So I mean, maybe this is a mundane question, but if it comes here from somewhere else, where does it come from in the first place, right? At some point, it has to bottom out somewhere. It has to arrive, even if it's not Earth. It has to be somewhere. Where do you think it comes from outside that would have had a longer history than we have available here?
So in the universe, there are a lot of so-called rogue planets. So these are planets that are lost from their solar systems. And they are traveling in space, and they can come long distances within the galaxy, and they can become absorbed within another planet system. And they are large, and because they are large, bacterial life can be there inside, even without any solar radiation, because it may live specifically on chemosynthesis, and the temperature can be sustained by nuclear reactions there, low-level nuclear reactions.